Greetings from the 85th Cold Spring Harbor Symposium on Quantitative Biology. This meeting is being held virtually this year because of restrictions from COVID, but the title is Biological Timekeeping. So I'm Brian Ray. I'm a senior editor at Science Magazine, published by the American Association for the Advancement of Science. And I've had the pleasure of attending the meeting and learning about how biological timing mechanisms are so important in controlling physiology and also health of organisms. And the organizers gave me the opportunity to sit down with some of the participants in the meeting and discuss some of the most exciting results that we heard about what's new in understanding of biological timekeeping. And so with me today is Maria Robles, who is from the Ludwig Maximilians University in Munich, Germany. And she is going to help explain some of her work. But before we started that, Maria, I thought I'd ask you, so how did you get your interest in chronobiology? This is kind of a particular part of biology that involves study of some things that not everybody's aware of. And so what attracted you to that part of the world to do your research?
Well, thank you for the interview and thank you for the questions. So I guess if I'm honest, I have to say that it was basically by chance. I did my PhD in immunology, looking at cell death, programmed cell death of apoptosis in the immune system. And I was exposed to international meetings, and I thought the immunology field was very established as an old field. And I wanted to move to do something different and kind of like a very different topic, and also something maybe less study. And then I look for personal reasons. I was in Boston. I moved to Boston and I looked in different places, and I found an interview with Charles White, and he explained me the topic, and I was fascinated because I never read anything about chronobiology at that time. So it was some years ago. It was the early days of chronobiology, and there were so many open questions. And I thought the topics was very exciting, and there was so much to learn, and I was hooked up immediately. And I have to say, if I can add something, the first meeting I attended, I loved the community. So it was such a friendly community, and it still is like this. And I love to be in meetings, and I love to see some of my colleagues now. And it's one of the parts that in addition to the biology and the topic, it's a pleasure to have this community of scientists. I have to say this.
Great. Yeah, so sometimes, like a lot of cases, it's contact with an individual, right? And this person's passion for a topic can really light that in someone else. That's interesting. Okay, so at this meeting, we heard a lot about circadian control of gene regulation. And of course, that's one of the huge focuses of the field, because the clock mechanism, the actual proteins that make up the biological clock in organisms like us, are transcription factors. And so they have this output that controls gene regulation in a daily cycle. But what you have emphasized is that it's not just gene regulation that's changing on a daily basis, but also there are changes in the abundance of mRNA, the abundance of proteins. And then you go on to show that there are changes in the post-translational modification of those proteins. And in particular, you study one of the things that I've been most interested in, which is the covalent modification of proteins. So the changes in phosphorylation of proteins, which is a chemical change in the protein that actually changes its activity. And this is a way that cells use to control circuits and activity of biochemical processes that actually enable them to regulate things and keep things going properly. So it's only recently that we've been able to measure across the whole spectrum of proteins these individual modifications of amino acids by phosphorylation. So you've brought a very sophisticated technique to understanding how these timing mechanisms are changing. You mentioned some recent work also that you're doing where you've developed software that allows you to assemble information on how these proteins are arranged in regulatory circuits and how components of the cell are changing in abundance during daily cycles, and allows you to put this information into a program and process it, so you can see how the timing changes occur and maybe infer from those how a particular phosphorylation event, for instance, might be changing the activity of a protein that's an enzyme and then producing a product that changes how the cell works. And so I think that's really interesting because we're now inundated with masses of data in the field of biology, where sometimes people stare at the data and say, what does this mean? But you've gave an example where you work yourself into that and come out and can explain, you can infer something that we didn't know before from looking at that. Can you kind of briefly tell us how you did that and how that works?
Yes, so I think the main motivation for me was also to be staring many hours at the data,
Yeah.
high throughput data that we are analyzing. And sometimes we have also very defined areas of multi-omics that we look at, but it's very difficult to integrate them, right? So with this software, we're trying to really take already published data and try to integrate it, in order to look at functional nodes that is regulating, so coming from the circadian, from the gene expression to protein abundant changes to phosphorylation that changes that most likely change the protein function, to the metabolic dynamics, and taking actually all of these omics from the same tissue and looking at temporal changes. We wanted to infer, because we know that the clock regulates metabolism and we have these metabolic changes, metabolic dynamics. We wanted to infer which step of all the way from gene expression to phosphorylation changes are really the main dominant functional steps here. And then we integrate all of these omics, not only in a steady state, but in a temporal manner, to look at cycles at the gene expression level of the mRNA, protein, and phosphorylation, integrating this with metabolic networks. So actually you can create these reactions knowing the metabolite that is the substrate and metabolite that is the product, and you can really make this relationship and say, okay, this reaction is dynamic, so it's changing because we have the level of the substrate and the product. And by looking at the dynamic reactions and looking at the enzyme that is mediating this reaction, we try to understand if this enzyme might be changing, because we know that the reaction is dynamic, if the activity of the enzyme is changing based in the mRNA that is also changing the protein level of the phosphorylation. And when we integrate all of these quantitative values into these reactions, what we saw is that the main data or high throughput data that explains the dynamic reactions are the levels of phosphorylations of the enzymes. It's not in some ways not surprise, because we know that protein activity is highly dynamic based on the phosphorylation levels of the proteins, as you mentioned at the beginning. And but nobody really had integrated all these time-dependent or temporal collections of all these omics data. Try to separate the quantitative values to really at the reaction level, what is really explained in the dynamic reaction at the metabolic level, right? And this is basically where we integrate all this quantitative data, and basically to see that the phosphorylation levels are the main quantitative data that will explain these dynamic reactions. And then you can actually have in this, you can have then not only dynamics, but also looking at the phase. And you can look at the phase. If you have more or less phosphorylation of this enzyme, you can predict when this enzyme will be activated, because phosphorylation can be an activated marker, but also an inhibitor marker, right? And by looking at the levels of the substrate and the product and the levels of the phosphorylation, and these relationships allow us to establish which phosphorylations might be driving the activity of the enzyme versus those phosphorylations that might inhibit the activity of the enzymes.
Right. So you gave an example. So you saw an activated enzyme, and as the phosphorylation of that enzyme went up, the amount of the product of that
Was going
reaction
up.
went up, but then the substrate that was being used in that was going down, just in the opposite phase. And so
Yes.
this timing information suddenly showed that relation of those three entities in that mass of data. It was very nice. All right. Well, you also published a pair of very impressive papers in Science Magazine recently that showed how phosphorylation changes during the daily cycle at synaptosomes. And so synaptosomes are these places where neurons actually contact one another. And so you have a method where you can isolate that particular part of the cells from the brain and look at these contact points where the neurons are actually talking to one another. And when you did that, you found some very interesting things about what's going on with phosphorylation in the brain. And can you tell us a little bit about that?
Yes. Well, this was a great collaboration with the group of Steve Brown in Zurich, in the University of Zurich. And actually, the story came from the transcriptional point of view, because Steve's question, the first question was try to understand if transcriptional dynamics, as we know, also driven by the clock, are also establishing different levels of mRNA at the synapses, because you have transcription, of course, in the nucleus. The mRNAs go to the cytoplasm, but when you're looking at synapses, sometimes the synaptoneurosome or the synaptic contact versus the nucleus are really far apart because the axons are very long. So the question was, do you need to have mRNAs to go to the synapses to really be producing proteins locally? Because then it happens very quickly. So you will not have to transport, after a certain cue to transport the mRNA or to produce the protein and move it to the synapses. So this was the first question. And we found, actually, that I'm moving from the phosphorylation, that we found that the mRNAs move to the synapses, and there they accumulate and produce protein after being challenged, the synapses from activity. So after synaptic activity, these mRNAs translate into proteins. But also we found that the degree of phosphorylation changes when we look at 24 hours are way higher than proteins. So, and back to what is the relevance of phosphorylations, I think, as you said, these modifications in proteins can be done very quickly. So translation takes some time, right? So even if you have the mRNA to produce the protein, you need some time to produce the protein, and the protein needs to produce the output of the function. So, but by modifying proteins and modifying the activity with these covalence modifications, we think this can be a very quick response. And these phosphorylations can really make very easy the system, in this case, the synaptoneurosome, to adapt to very small, fast changes. For example, synaptic activity or neurotransmitter signaling pathways downstream of the neurotransmitter receptor. So we found that basically half of the proteins that are phosphorylated in the synaptoneurosomes have phosphorylations that change with a periodicity of 24 hours. So we think phosphorylation is a very important post-translational modification in synapses, most likely to adapt to these very fast changes that synaptics are encountering across the day.
Right. So you've shown that these changes that are going on a daily basis are very dramatic, and they're particularly so when you drill down in space to these particular regions, right? That this is very specific. Some of the things that are lost in a global look at what's going on are revealed when you have this ability to hone in and on this contact point where the neurons are working, in which you might expect to be a very active place. And then there are tremendously large changes in the circadian changes in the amounts and modifications of these proteins. So when you saw these changes in phosphorylation at the synapses, there could be two things going on, right? We have the circadian clock that's cycling through a 24-hour phase, but we also have the sleep-wake cycle the animal that's changing what's going on in the brain. And so you considered whether is it actually sleep that's influencing some of these changes that you see at the synaptosomes, and investigated what happens if you put those animals under sleep stress or deprivation of sleep. And so tell us what you found there.
Yeah, so I think the question was actually coming from the data. So we look first at cycles, and we saw a lot of cycles, but then when we look at when these cycles are happening or where is the maximums of these phosphorylations, we immediately see that most of the phosphorylations have divided very much into clusters. One of these transitions from the sleep phase to the wake phase, and the other one, in antiphase, actually, when the mice will go from a wake phase to a sleep phase. This was what they gave us the hint. Maybe, it's not necessarily all circadian driven, but sleep, the change in these two states between sleep and wake and wake to sleep might drive these changes. And indeed, this is what we, when we study further by sleeping depriving, so we made a sleep deprivation protocol. And we look at now, if now you don't allow the mice to sleep, you will break these cycles of sleep and wake between day and night. And whatever is remaining should be circadian, because the circadian mechanisms is not changing dramatically after sleep deprivation. And actually what we did find is that if we break these sleep-wake cycles, all the rhythms of protein and phosphorylations are abrogated. So the main rhythms of translation and protein abundance and phosphorylation changes and signaling pathways are mainly driven by sleep-wake cycles. And again, I think biologically speaking, maybe also makes sense because these sleep and wake cycles are very fast states, right? So the mice are going to be awake, and then this has to be very fast. You need to mechanisms to respond quickly to this. And we think phosphorylations is a perfect mechanism to control this, because it can be very quick and then can really quickly change the activity status of the proteins. And basically, within the synaptic function, with the protein and phosphorylation status as proxy, is mainly driven by synaptic, by sleep and wake cycles rather than circadian mechanisms.
Yeah, I thought that was really interesting, because here you start by just characterizing what do we see in these synaptosomes, what do we see in the biochemical changes there, but it leads you to something that sounds like it might be answering a really fundamental question, which is what is this process of sleep doing that is so beneficial? We know that people have to sleep. We know that sleep deprivation is bad, but what we really know very little of what is the actual physiology, the biochemistry that's being replenished in a sleep process. And here you point us at this interesting process of protein phosphorylation and changes in enzyme activity that seems like it opens up a whole little field of itself, a way to maybe explain some of these mysteries of sleep.
Yeah.
Okay, well, thank you so much, Maria, for taking the time. Really impressive presentation at the meeting, and we appreciate your taking the time to explain it to us today.
Well, thank you very much. The pleasure is mine, and I hope everybody enjoyed the meeting. It was a great meeting. Luckily, we could see each other here via Zoom, but it would have been much better in presence. But I think we managed. Yeah, thank you, Brian, for interviewing and talking about our data. So we are very happy about this, and yeah, and College Spring Harbor to host us and give us the opportunity.
All right, thank you.
Yeah, thank you very much. Yeah, bye-bye.
Bye.